I'm going to give a short dedication to Jay Eastman.
Yes.
Because he was, after all, part of the university.
Amen. No, actually, that's very fitting. Very, very fitting. A big hole. I mean, sometime over dinner, actually, I tell you, he is what made the laser lab happen. I don't know if you ever heard that story, but he was a project manager on it, and they, all the scientists said they needed $80 million. And don't even bother. We need $8 million. Well, Moshe had raised 15. And so Jay went, yeah, okay. Basically went ahead and said, what can you do for 15? And under his leadership, they actually got it built.
Wow.
Okay, let's go ahead and get started now. I see we are just 2 cookies away from being able to have it. It's my pleasure to introduce Milind Rajadhyaksha. Dr. Rajadhyaksha completed his bachelor's from the Indian Institute of Technology in Mumbai in 1985. And at that point, he went to Purdue University to the Applied Optics Laboratory, which is actually a relatively famous laboratory from the Institute of Optics perspective, because that's the same group that Balamanian, a faculty of the Institute, received his PhD as well. Milind completed his PhD in 1991 and then proceeded on to appointment as a research scientist at the Wellman Laboratories of Photomedicine at Harvard Medical School. And he held an appointment at Harvard and at Wellman until 2002, I believe. Mm-hmm. But along the way, he's done a lot of things. I had the opportunity to work with Milind kind of starting around 1995 when the company that I was involved with, Lucid, entered into a relationship with Wellman, in which we commercialized the design that Milind had come up with Rob Webb, now called the Vivoscope. And from 1997 to 2002, he was an employee of Lucid. And then in 2002, he joined the faculty at Memorial Sloan Kettering. He holds appointments at Weill, Cornell, continues research with Northeastern University, and is kind of a person that goes around the world helping to promote the optical application of in vivo and ex vivo imaging. So, Nolan?
Great. Can you all hear me?
Yes.
Perfect. So first of all, Jim, thank you very much for the kind introduction, and thank you for asking me to visit and to speak to all of you about my work. And it's great to be back in Rochester. The last time that I was in town was, I think, in mid-2019. And then, of course, all of us had to live through the pandemic years. So it's really nice to be back in town. It's great to see my good friend and colleague, Jim, once again in person, not on Zoom. You know, he looks so much better in person than on Zoom. And good to really catch up with many of my friends and colleagues. I have many of them in this town. Hello, everyone. A very good afternoon to all of you, and thank you all for coming. I'm going to speak to you about reflectance confocal microscopy of skin cancers. And I'll give you a sense for the technology that we've created, clinical research that has taken place over the last now more than a couple of decades, and now the early impact of this imaging approach on patient care. So I'll give you more of a bird's eye view of the field and give you a sense for what has happened over the last 20 to 30 years and what's happening today and what may happen in the future. As some of us in this room are well aware, confocal microscopy of skin and imaging of skin cancer started in the late 1980s, early 1990s, before some of you were born. That's about more than 30 years ago now. Starting with the development of new devices, new instrumentation, new technologies. And since then, this field has evolved into what has today become a rather lively, a rather active, a rather vibrant area of work. So much so that some of our imaging work has progressed in recent years all the way into the clinic and is being used routinely at the bedside to help detect skin cancers and to help guide patient care in many different clinical settings worldwide. The very first paper on confocal microscopy of human skin in vivo was published back in the year 1991 by J.B. Jester, who is a well-known ophthalmologist in our field of biomedical optics. Then came a string of papers from a famous pair of physicists, Pierre Carcouf and Jean-Luc Levesque. Now, much of this early work was with white light confocal microscopy, meaning that their confocal microscopes used white light sources for illumination. Then came two other groups. We were the third group to enter this fray, and we started building microscopes using lasers for illumination. So our work was laser-based confocal microscopy. And by we, I'm referring to my two mentors, Rob Webb, who was a very well-known physicist in our field, and Rox Sanderson, who's a very well-known dermatologist. And around the same time, my good friend and colleague Jim Zivisland, along with Jay Eastman, had started building handheld confocal microscopes for imaging of skin. Okay, before I go on any further, let me go ahead with my daily confession and get this out of the way. A couple of my microscopes have been commercialized and translated by this company, Caliber Imaging and Diagnostics, which was known formerly as Lucid Incorporated. This is a local company. This is literally a couple of miles from campus. And a long time ago, I worked for the company for a few years, back when Jim and I were together at the company. In nanoslice. And I wish to give this talk today in honor of Jay Eastman and in honor of his pioneering work and what's going to be now his legacy. Many of you here know of Jay Eastman. You knew Jay because he was one of your colleagues at this university. Many of us know Jay Eastman as the founder and the CEO of Lucid Incorporated, which is now known as Caliber Imaging and Diagnostics. Jay's company is the company that has given us reflectance and fluorescence confocal microscopes for skin cancer diagnostics. And as Jim mentioned, the product, the commercial product microscope is called the vivoscope. So a long time ago, when I was with the company, when I was with Lucid for a few years, I'd worked very closely with Jay. And to me, he was just a wonderful boss and a supervisor. He was a great colleague and a mentor, and eventually, of course, a dear friend. So for me now, it's going to be a tremendous pleasure and a tremendous privilege to continue my work in honor of Jay's work and to help carry forward his legacy as some of us in this field continue to persevere to build microscopes and to build imaging tools and devices to help address the problems of detecting and diagnosing and treatment of cancers in many different settings. All right, so why do we care? Why do we care about non-invasive approaches for the detection and the diagnosis of skin cancers? Well, it turns out that among all the cancers which occur today, skin cancers occur with among the highest rates of incidence and high prevalence, not only in this country, but also in many other countries around the world. Every year, if you look at the numbers, every year, somewhere between 5 to 6 million new cases are detected in this country alone, for which millions of biopsies are being performed. Now, depending on the setting, depending on the patient population characteristics, depending on the skills of the clinicians, depending on the resources available or not available, it turns out that large numbers of these biopsies, 60%, 70%, in some settings, more than 90% of these biopsies turn out to show skin that's essentially benign, meaning that many of these biopsies should not have been done or possibly should have been avoided in the first place. So among children in the pediatric patient population, the number is easily a couple of hundred biopsies of benign skin conditions for every detected skin cancer. And among adults in the adult patient population, the number is easily few biopsies to a few tens of biopsies of benign skin conditions for every detected malignancy or for every detected cancer. And the cost of doing all of these diagnostic biopsies is easily a few $1 billion every year to our US healthcare system. So the question that we began asking ourselves back in the day was quite obviously, can we come up with better ways to detect and diagnose skin cancers? Could we do this perhaps directly on the patient? Could we do this non-invasively while trying to reduce the need for a biopsy? Could we do this in real time at the bedside, hopefully at a cost that would be economically viable, both for the patients as well as for the healthcare system? And the question that some of us began exploring, Jim and I, as well as some of our other colleagues, was whether we could do this with an approach such as confocal microscopy, a high-resolution microscopic imaging approach that shows patterns of cells and patterns of nuclei and the kind of detail that pathologists look for in their biopsies and in their frozen and their fixed pathology sections. And if you look outside this country, particularly in some parts of Europe, in UK, in Australia, you're looking at about another close to 1 million new cases being detected every year. So it's truly a global problem. Now, visual examination and dermoscopy are routinely performed in the clinic. Think of a dermoscope as a magnifying glass, if you will, something like about 2x to 3x to 4x magnification, somewhere between 5 to 10 microns of resolution. Now, in the hands of a highly skilled physician, a highly skilled clinician, a highly skilled dermatologist, dermoscopy can provide high sensitivity for detecting the presence of a particular skin condition and high specificity for detecting the absence of a particular skin condition. But in general, the specificity tends to be variable, and it can be quite low in some settings. And especially true for lesions or skin conditions which lack distinct patterns and distinct features. So if you look at, for example, these two skin conditions, these two images, you can't tell whether this is simply just normal skin or whether there might be something bad happening below the skin surface, whether there might be something potentially malignant. So dermoscopy lets you look at skin from the surface, but not below the skin surface. And that results in a large number of false positives, which then leads to biopsies of benign skin conditions. So with dermoscopy, it's a form of conventional volumetric microscopy, if you will, which means you illuminate, you image, you detect light from the entire volume of tissue within a skin lesion. So within the volume, you can look at patterns of pigmentation, you can look at patterns of morphology, you can look at patterns of blood vessels and blood flow, but you cannot look below the skin surface to look at any individual layer of cells or any individual layer of nuclei. So all of the patterns of cells and nuclei that are in the skin, they remain hidden below the skin surface, which is why oftentimes beyond dermoscopy, one then proceeds to performing a biopsy. We take the piece of biopsied skin and we slice it into thin sections. We stain those with purple and pink colored stains called hematoxylin and eosin, and we look at those sections with a microscope. And that's how we can start looking at patterns of cells and patterns of nuclei below the skin surface. And this, of course, is the traditional process of biopsy and microscopy and pathology. But with the confocal microscope, we can do all of that non-invasively. We can perform what is known as non-invasive optical sectioning, meaning that we can image thin sections or thin planes below the skin surface without the need for a biopsy and without the need to cut tissue into thin slices. And these are example images showing you normal human epidermis, which is on the upper left-hand side. You can see basal cells on the upper right-hand side, and example images of a melanoma skin cancer and a basal cell carcinoma. All right, so at this point, you must be asking yourselves, what exactly is a confocal microscope and how does it work? So just a quick show of hands, how many of you here are familiar with confocal microscopy? Wow, quite a few of them. Very cool, this makes my life easy. But there are a few of you who seem to be not familiar, and so for you, I'll give you a quick tutorial. Now, in this room, we all love optics, right? We all love the theory of optics. We all love the theory of imaging. But I promise you, I'll keep this very simple, very conceptual, no math, no theory, no equations, at least for this hour. A confocal microscope is made up of a point source of light with which we illuminate a small spot of light or a voxel within tissue or within skin. For those of you who are familiar with confocal microscopy or with the theory of imaging, you'll know that this is the illumination point spread function. Light which is scattered back from the voxel is collected through a small pinhole onto a detector, and that will then produce for you a pixel. So what you're doing by using a pinhole in front of the detector is collecting singly back scattered in focus light coming back from the tissue. Light which has gone in and has become multiply scattered, which means light which has bounced around quite a bit and has become defocused. That light, which I've shown here with these dotted lines, red and green dotted lines, this light is rejected or spatially filtered by that pinhole in front of the detector, okay? Now think about it. If you were to remove the pinhole and collect all of the light, what would you have? What do you have? You'll have a dermoscope, right? Because that's what exactly a dermoscope does or dermoscopy does. So in the confocal configuration, it's a very clever approach for detecting only singly backscattered light coming back from the illuminated voxel, looking at only in-focus light. A very, very clever idea, but oftentimes not very useful because one needs to look at large areas or large volumes of tissue. Particularly physicians and pathologists need to look at large areas of skin or tissue to be able to arrive at a proper diagnosis. So what can we do? Well, what we do is we scan the voxel in 2 dimensions. And this, by the way, is my rather pathetic attempt at PowerPoint animation. This took me a good 2 or 3 hours to figure out how to do. This was, many years ago, at which point I decided to stop and get on with the rest of my life and let you imagine what really happens. But imagine the voxel being scanned in 2 dimensions. And do I have a laser pointer? Or maybe not. Okay. So imagine, if I had a laser pointer, I'd kind of sweep. Oh, I think Honey has. Oh, perfect. Great. Thank you.
So
It is on now.
Yep, there you go.
Oh, green. Oh, green, even better. All right. So just to show you scanning. So what we're doing is we take the voxel and then we scan it back and forth. Okay. So what you're doing is you're creating a plane of voxels within skin or within tissue. And of course, while we and this happens really quick, by the way, this happens really fast. And while we're doing all of the scanning, we keep getting back light that's all multiply scattered and is all out of focus light. We continue to reject all of that light. And as the voxel is scanned, as you move back and forth, you collect light from each location. Each of those locations will produce for you a pixel consisting of singly backscattered light. And what you now have is an array of pixels that will produce an image, much like you have an array of pixels in your cell phone camera or in your laptop monitor, okay? So as you can see, with this approach, what we can do is we can, by using a voxel for illumination, a pinhole for detection, and by scanning, what we can do now is image a thin plane or a thin slice or a thin section within skin or within tissue. And we can do that non-invasively without the need for a biopsy and without having to physically cut skin into thin slices. Does that make sense to everyone? Good. So what we're doing, so this is known as non-invasive optical sectioning with a confocal microscope. It's a very cool idea, very cool configuration.
How deep can you go?
That depends on the illumination wavelength that you might use and also depends on the properties of the tissue. Typically, with visible wavelengths, we can go down to a depth of 50 to 100 microns. If you use longer wavelengths like near IR wavelengths, you can go down to about 200, 300 microns. We do imaging with very high resolution, which is why we don't image too deep. But if you were to also do imaging with lower resolution, which is what happens with OCT imaging, which I'll talk about in a short while, then you can go deeper. Depends on the illumination, numerical aperture, and tissue conditions. But in general, it's imaging in the shallow layers of tissue. We don't image too deep. It's optical imaging. Any other questions? Any other comments at this point? So back to dermoscopy, which is a form of volumetric microscopy, meaning that we don't have any optical sectioning. The depth of field is typically tens to hundreds of microns, and so we don't reject any out of focus light. But with a confocal microscope, as you've seen, we can perform non-invasive optical sectioning. And we can begin to look at patterns of cells and patterns of nuclei below the skin surface, okay? So what you're seeing here are example images showing you normal human epidermis. These are basal cells in human skin and example images of melanoma, skin cancer, and basal cell carcinoma. This shows you the original benchtop confocal microscope that I put together. This was many, many, many years ago. My friends like to remind me that this was all now back in the last millennium. And this was back when I was a postdoctoral research fellow in Boston. What we have in this setup is a collimated laser beam, which behaves as a point source at infinity. So there's your point source of illumination. That light, that beam is focused through a microscope objective lens into a voxel. And that's the voxel that you create within your skin or within tissue. That voxel is scanned in 2 dimensions with devices called optical scanners. In our case, we used a spinning polygon mirror, and we have an oscillating galvanometric mirror. And there are many different ways to perform beam scanning. And as the voxel is scanned in 2 dimensions within tissue, we collect light from each location, and the light will basically retrace the way it came. It comes up to a beam splitter, at which point it separates from the incoming light and is focused through a pinhole onto a detector. And so there's our pinhole, which is optically conjugate to the voxel that makes this setup confocal. I thought I might amuse you and entertain you by showing you some of my very first images of skin, which are just plain awful. It turns out that the images that you see on the top came from a piece of rabbit skin, and the images below are the skin on my arm. And it took us a good amount of time to figure out how to make these images better. Jim will remember, Jim is smiling. Jim will remember many, many late nights we spent in the lab, chasing down speckle noise and all other forms of noise and trying to figure out how to get better optical sectioning and good point spread functions and better alignment. And you know what happens in the lab? Good things happen only after midnight. Nothing good happens between 8 a.m. and about 11 p.m., right, Jim? So much of what much of the imaging that we did that turned out to be good all happened in the very late hours at night. But that's what makes it fun, right? To do research and to work. Measurements of axial response function, which gives you a sense for how detected signal falls off as you move away from the focal plane or as you move away from the optical section. The full width at half maximum of such response functions gives you a measure of optical sectioning. And that depends on the numerical aperture, the wavelength, the pinhole conditions. And then the tails of the function gives you a measure of how much light you might collect as you move away from the focal plane. And that's all of the multiply scattered out of focus light, some of which will get through your pinhole and will give you background noise and will give you reduced contrast. So with a well set up, well configured, well designed, well aligned optical confocal microscope, and by the way, proper alignment only happens in the hands of graduate students, never their PIs or their fellows. So if you have a really nicely set up confocal microscope, you can expect a rather sharp axial response function, which is I've shown here in blue. And so that's the kind of sharp imaging that you can expect in the upper layers of skin known as the epidermis. But as you go through all of the epidermis and you start going into the deeper layers of skin, which are called the dermis, the axial response function begins to degrade, as shown by the red plot here. You can see how the optical sectioning becomes worse and the tails become wider, meaning that you start collecting more and more light that is away from the focal plane, and that will reduce contrast and give you background noise. And that happens because as we go from the upper layers into the deeper layers, there are effects of scattering and aberrations that begin to build up and affect our axial response function. And Jim has spent quite a bit of time looking at the effects of aberrations on the imaging performance of our confocal microscopes. A similar story if you were to make measurements of an edge spread function, which gives you a sense for how lateral resolution will vary. And so if you were to image a sharp edge, you can expect a sharp edge spread function. But as you start imaging through a piece of human epidermis, the edge starts appearing blurry, and you can see how the edge spread function also begins to degrade. And that gives you a sense for how your lateral resolution begins to degrade as you start imaging deeper in tissue. So back to my setup that I'd put together. This occupied something like 2 feet by 4 feet of optical bench space. There's a whole rack of electronics, which you sort of see in this figure. And that's how we got started. This was a microscope that did not move. So we had to physically bring volunteers and patients, especially some of my friends at the lab, unsuspecting volunteers, and shove them against the microscope and hold them still while we did some of our early imaging work. But that's how any new device or any new technology makes a beginning. And this is now what we have in the clinic. This is one of our fancy mosaicing confocal microscopes. As I mentioned to you earlier, this technology, this microscope, was commercialized by Jay Eastman and Jim Zavislan and Bill Fox, one of our colleagues at the company. And this was back in the day when Jim and I worked together at the company, at Lucid. This microscope has been around now for 2 decades, has been the workhorse in the field, has been used in a very large number of clinical studies and trials, some of which I'll talk about in a few minutes. And it's also now being used routinely at the bedside to detect skin cancers and to help guide patient care in a number of different settings worldwide. By moving the objective lens towards and away from skin, we can collect stacks of images, and we can look at skin or tissue in depth. As well as, at any given depth, we can collect a 2-dimensional sequence of images, which we can then register and stitch together into a mosaic to display a larger area or a larger field of view. Very necessary because clinicians and pathologists always need to look at large areas of tissue to be able to arrive at a proper diagnosis. So our imaging is typically with 30x magnification that shows a rather small field of view, but we can produce mosaics that show up to several millimeters by several millimeters of area. And of course, one can zoom into such mosaics to look at skin with different magnifications in different fields of view while maintaining resolution. Okay, so this process of mosaicing really mimics the process by which a pathologist looks at pathology, which typically starts with 2x magnification that shows 10 millimeters by 10 millimeters of field of view. And what they wish to do always is to look at patterns of cells, patterns of nuclei, patterns of blood vessels, patterns of morphology, patterns. They don't wish to look at individual cells and nuclei to begin with. And then whenever they spot areas of suspicion or areas of possible malignancy, they zoom in with typically 4x magnification and with 10x magnification to more closely look at areas of suspicion. This happens to show you a basal cell carcinoma in skin. So we can mimic this approach. We can mimic this process with our mosaicing. So I'll show you some example mosaics. This came from one of our vivo scopes. This one shows you a melanocytic skin lesion, which is a clinical term for moles, right? All of us have moles on our skin, pinkish, purplish, brownish, blackish spots. And in this particular mosaic, what you're seeing are what are called as these ring patterns of basal cells sitting at the dermal epidermal junction, sitting at the junction between the epidermis above and the dermis below. In this particular mosaic, you can see the ring pattern appears quite degraded. This looks rather cruddy. This looks awful. This happens to show you a melanoma skin cancer. And in this particular mosaic, what you're seeing are these darkish, grayish, hyporeflective nest of tumors, what are called as nodules of tumors. This happens to show you a nodular type of basal cell carcinoma. We also have a handheld confocal microscope in the clinic, very useful for acquiring videos, which we can then process into video mosaics, which is very useful for mapping margins or boundaries of skin lesions, which can help guide surgery and can help guide treatment of these lesions. And just an example of a completely mapped margin of a lentigo-malignant melanoma, that's a particular type of melanoma. And in this melanoma, typically you have margins that are circular, and then you have cancer that spread in radial fashions like the roots of a tree. And if we were to zoom into this video mosaic, there's a lot of detail here at the level of patterns of cells and nuclei and morphology that can help surgeons detect margins, which can then help them guide surgery. All right, so this brings me to the end of the first piece of my talk, which was a quick introduction to the technology. I'll now switch gears. I'll switch topics and talk about imaging. I'll show you images, and we'll talk about some of our clinical work. But let's take a break for 2 minutes and see if anyone has any questions, any comments, any feedback, any opinions, any perspectives. Oh, go ahead. Yeah.
The commercial device that you showed, then the laser that you use is the near infrared, right?
Yeah, 830 nanometers.
830 nanometers.
Yeah.
That's where the scattering of the skin is also so little.
Right. Yeah, so we've tried wavelengths between visible going up to about 1064 nanometers, because those are the wavelengths that the scattering does decrease, of course, as you go to the longer wavelengths. And also beyond about 1300, there's absorption by water that begins to increase. So there's that window that we always use for imaging. So 830 is a good wavelength, 900, 1064 is another good wavelength that goes a little bit deeper. Yeah. And then, of course, I have colleagues and, looking at using multiple wavelengths all at the same time so you can image at multiple depths, for example. Yeah. Any other questions? Okay, so moving on. Thank you.
You can hear me or
The
not?
pathology is based on looking at sections oriented perpendicular to the skin surface. But the laws of optics are such that with a confocal microscope, our best imaging is in planes oriented parallel to the skin surface, what is known as the en-face orientation. So we can visualize skin in non-invasive en-face optical sections, okay? Our imaging is based on the detection of singly backscattered light from the optical sections. Remember that pinhole in front of the detector? We collect only singly backscattered in-focus light from the illuminated voxel. And we get contrast due to refractive index variations between all of the different structures that are present in the cytoplasm. The pigment melanin, the protein keratin, organelles like mitochondria. And so what we get is endogenous backscattered contrast. It's not truly reflectance. I call it reflectance for convenience, but it's really backscattered contrast. What this means is that we rely on the natural backscatter or the natural reflectance from skin. We don't use any contrast agents. We can't use hematoxylate or eosin. We can't use most of the stains used by pathologists because they're all toxic to skin and they would be toxic to the patient. So in backscattered contrast, in reflectance contrast, nuclei appear dark because of weak backscatter from all of the chromatin, but the surrounding cytoplasm appears bright because of strong backscatter from all of the different structures present in the cytoplasm. And Jim, could we load the lights? No, right here. Oh, go ahead. And load them completely. Oh, do not go to sleep, anyone. My eyes are on you. The pigment melanin has a high index of refraction relative to the surrounding epidermis, and melanin is organized into it's packaged into tiny granules called melanosomes, which are strong backscatterers of light. With the result that melanocytic skin lesions, lesions which contain melanin and or melanosomes, pigmented skin lesions, melanomas, as well as amelanotic melanomas, all of these different types of lesions image with exceedingly strong brightness and strong contrast. So in our case, melanin behaves as a stain, if you will, a natural stain within skin. Okay, so on the left here are images showing you the different layers that make up the epidermis. Stratum corneum, granular cell layers, spinal cell layers, then you have a single layer of basal cells. And then below the epidermis, you have what's called as the dermis, which is mostly connective tissue. So you have what's known as the papillary dermis. You see blood flow because there are tiny capillaries that come up to below the skin surface. And then going a little bit deeper, you can see what's called as the reticular dermis. And with our imaging, with the wavelengths that we use, we can typically image to a depth of about 100 to 150 microns, which means down to about the papillary dermis. So what I'll do now is show you very quickly a video to give you a sense for how normal skin appears when we image with one of our microscopes. What you'll see are cobblestone patterns of dark nuclei within honeycomb patterns of bright cells, and that's the description that was coined by one of our colleagues in dermatology soon after they started imaging and looking at skin with our confocal microscopes, okay? So honeycomb patterns of cells, cobblestone patterns of dark nuclei. So there's the stratum corneum. Granular cell layers, look at that honeycomb pattern that you'll see here. Spinal cell layers, rings of basal cells opening up, and then below the basal cell layer, you can see thin slivers of blood flow. If you can tell, you'll see tiny slivers of motion, that's blood. And going a little bit deeper, you'll see some of the deeper collagen and connective tissue in the papillary dermis. Let me show you that one more time. Whoops. Stratum corneum, granular cell layers, honeycomb patterns of cells, spinous cell layers. That's a sweat duct, by the way, that's opening up. Rings of basal cells, and then below the basal cell layer, thin slivers of blood flow. You can see that right here. And then going a little bit deeper, you'll see connective tissue in the papillary dermis. That's about somewhere here or here. So the limitations with this imaging approach are that the depth of imaging is limited to the papillary dermis. That's about 100 to 200 microns, more like 150 microns. Nonetheless, this depth does allow our clinicians to routinely image and observe and examine the dermal epidermal junction, and especially the basal cell layer. So they can look at the lower spinous layers, the basal layer, and the underlying papillary dermis. And that's a region that they always wish to look at, because in the basal layer, at that junction is where most of skin disease and cancer will originate. So typically disease starts in the basal layer, and then when it becomes cancer, it might spread laterally along the dermal epidermal junction, or the cancer may invade and start going deeper into the dermis. So that's the key region that we can image. Our contrast is grayscale. If you remember, our contrast is based on natural reflectance or backscatter from skin, with the result, and we use a single wavelength at the moment. So it's monochrome wavelength. So our images in grayscale contrast lack differential contrast for structures, meaning that we can't always distinguish between melanin versus keratin, between melanocytes versus other types of cells, and between some types of inflammatory infiltrates. Now that's on the engineering side. On the clinical side, there are some pitfalls for detecting some types of lesions and reading images of other types of lesions. And these are challenges faced by our colleagues in dermatology, our clinicians. And these include nodular some types of melanomas, which may be too deep or which may appear with not any distinct features or patterns, as well as some of the smaller micronodular and infiltrative types of basal cell carcinomas. These tend to be too small, and oftentimes they tend to be too deep. Nonetheless, despite these pitfalls and despite the engineering limitations, our images do present a rich amount of detail in terms of patterns of cells, patterns of nuclei, patterns of morphology, and you've seen some of these already. These are example images of melanoma and example images of basal cell carcinoma. With the result that our colleagues in dermatology have learned to read these images, and they're able to interpret these images, which then helps them guide diagnosis. With the result that there's been tremendous progress in translation over the last now more than a couple of decades. Now, like all of you, I'm an engineer by training, and I'm a researcher. And so for me, being involved in some of this work was just quite exciting. And it was just tremendous fun to watch all of this progress happen in the clinic in the hands of many of my colleagues in dermatology, not just in this country, but also in Europe, particularly in Spain and in Italy and in Germany, and also in Australia. Now, the upshot of many, many clinical studies and trials over the last more than 2 decades has been that reflectance confocal microscopy, if you combine that with dermoscopy, will give you about 90% sensitivity for detecting the presence of skin conditions or skin cancers, and about 60 to 70% specificity for detecting the absence of skin cancers. Now, it turns out that this specificity is 2 times higher than what you might get with dermoscopy alone. Remember, with dermoscopy, it varied between 30% to about 60, 70%. And as a result of the increase in specificity by 2 times, the number of false positives beyond dermoscopy has decreased by 50%. And what that means is that the number of biopsied benign lesions per detected cancer has now dropped by about 50% with the use of reflectance confocal microscopy beyond routine dermoscopy. So starting in the year 2013, which is now a decade ago, many of my colleagues started combining dermoscopy with reflectance confocal microscopy to begin to triage lesions into those that appear to be benign, and such lesions are now being spared from biopsies, and they use imaging to keep a watch on those lesions and monitor them on a routine basis. So that has resulted in a 50% drop in the number of false positives versus those lesions that appear to be suspicious. And of course, these lesions do proceed to being biopsied and being looked at with pathology. And so based on all of this work, which took more than 2 decades, 24 years to be quite exact, between the time that we started doing the work in 1992 to 2016, and it took a global village of colleagues and collaborators from all over the world to move this imaging forward into the clinic. But what happened in 2016, I'm happy to tell you, is that reimbursement codes were granted for reflectance confocal microscopy for imaging of skin. This is a big deal for any new technology or any new device, especially in the US healthcare system, because what this means is that your physician can perform reflectance confocal imaging on your skin and can bill for the procedure, and your insurance company, your insurance payer, will pay for the procedure. So getting reimbursement codes is a key step towards the acceptance and the adoption of any new device or any new technology or any new drug or any new procedure. So a big step forward for this technology, a big step forward for all of us in the field of biomedical optics and in dermatology, a big step forward for the company, and a big step forward for some of us at a personal level, for Jim, for me, and for some of us from Lucid. So what's happening now? Early adoption is in progress, as you might imagine. And the last I heard from my colleagues at Caliber, there are now about 1,000 clinics worldwide that are beginning to do reflectance confocal microscopy routinely to help detect skin cancers on patients and to help guide patient care. So what next? What do you think we should do next? Where can we go from here? Well, what comes after diagnosis, typically? Treatment. So as you can imagine, so our work over the last 3 decades has been focused on imaging to help guide diagnosis. But as you might imagine, new areas of work, new application, and what we're doing now is trying to figure out whether imaging can possibly help guide treatment. And what's actually turning out to be even more exciting is the possibility that imaging might help us combine diagnosis and treatment all in a single patient visit, what's being called by my colleagues as a one-stop shop patient care paradigm. And so I'll now change topics again and talk to you a little bit about some of our work in progress, which is on imaging to help guide treatment. But once again, let's take a break for 2 minutes and see if anyone has any questions. And please do wake up if you're napping. We're going to turn the lights on. Any questions, any comments? Yeah.
The RCM, for you make for the images, do you need to train the doctors to read these images, or because that's the deal with the pathology, and I know that the Rama that the doctors say, oh, we don't know this.
Yeah, we have the same challenge because much like they have to be, much like the pathologists are, get training to read pathology and the radiologists get training to read radiology, we've had to also provide training. So what has happened is that the first generation of clinicians who began working with us and started doing all the clinical studies became trained to read these images. And so they're now holding courses and they're giving lectures and they're conducting workshops to begin to train the next generation and the next generation.
And I told you are thinking about it, that to couple it with the AI pattern.
I'm glad you said that. We're coming to that, yes. We have begun some effort with machine learning. And so a long way to go. These images are not easy to deal with machine learning, but we'll talk about that. Yeah. Yes. Yeah, we do have motion to deal with. And so the way we do this is that we have our objective lenses, which actually are designed to make contact with the skin. So these lenses will image through not standard cover slips, which are typically 150 microns thin, but actually they've been designed to work through a millimeter thick window. And the window is integral with the objective lens, and everything sits on the skin and makes contact, and that helps. So that the two ways of doing it. One way of doing it is to actually physically attach a ring to the skin that we've been doing, and then the objective lens kind of locks into the ring. That's with one of our bigger microscopes. And then with the handheld, we don't have a ring, but the lens just sits on the skin, and you can apply gentle pressure to keep it still. So there's always a little bit of motion, but it's not too much. And an imaging is at higher speeds to account for motion artifact. Yes, sir. Oh, Surgivance? Yeah, I'm familiar with the company. Okay, so the question here has to do with whether we can colorize images and make them look purple and pink, similar to what pathologists are accustomed to looking at. And so we've done quite a bit of work looking at excised tissues. We can't use dyes or stains in people, not just yet, although there are now colleagues using machine learning approaches to see whether they can colorize images. But with excised tissue, biopsy tissue or tissue that's being removed from the patient, we can start using contrast agents. And so with a contrast agent that labels nuclear morphology, for example, in fluorescence, we can do imaging in fluorescence and look at nuclear morphology. And then you can take those images and then colorize those purple. You can then look at the same piece of tissue in reflectance contrast and see not nuclear morphology, but you can see cytoplasm and dermal morphology, and you can colorize that pink. And so what you're mimicking with your reflectance and your fluorescence imaging is staining with purple and pink colored stains. So some of that work has been commercialized by Caliber Imaging and Diagnostics. There's a company in New York City, Surgivance, that has come up with other ways to colorize tissue. This is an excised tissue, correct? Correct, yeah, this is all in vivo work. Yeah, and I do have a few slides on our ex vivo work, and since you brought it up at the end, I can just pull up my PowerPoint and show it to you. Yeah.
We have a question from the chat.
Yeah.
Does hyperkeratotic lesions impede with the squamous lesion detection?
Yes, for sure. Wherever there's too much keratin and there's hyperkeratotic situations, we don't image too deep. And so that is one of the limitations of reflectance confocal microscopy, or with most of our imaging techniques. Okay, all right. Any other questions? Yes.
You mentioned that how does that affect, like, I guess, the image quality or the image
Yeah, if you put too much pressure, you would end up deforming the skin. Yeah, so there's there's some skill involved in making gentle contact, which many of our students and fellows, and now in the clinic, the imaging personnel have learned to do. So yeah, one has to be a little bit careful. Should I keep going? Okay. All right. Yeah, can maybe just the front, not so much the back. Okay. Yeah. All right. So as you've seen, reflectance confocal microscopy, you can leave them. Reflectance confocal microscopy at the dermal epidermal junction helps guide diagnosis. But there still remains a need for imaging deeper to look at the deeper margins of skin lesions, to look at the depth, which can then help guide treatment. And so what we've been doing in recent years is combining reflectance confocal microscopy with another optical imaging modality called optical coherence tomography, another well-known imaging approach, or OCT imaging. So similar to confocal microscopy, OCT imaging also performs optical sectioning. So one can non-invasively image thin optical sections, albeit now we do imaging with lower numerical apertures. So we trade resolution for depth. And so we don't see patterns of cells and nuclei. We don't have that kind of resolution, but we can see the different layers that make up skin, such as the epidermis, the dermis, the dermal epidermal junction. We can typically go down to a depth of several hundred microns up to a millimeter. And these are example images of different types of skin conditions. So for OCT imaging, we use a broadband source of illumination, and interference between a reference set of waves and a sample or a probing set of waves is what gives us what's called as a coherence gate. That's our optical section within skin. Okay, there's the optical section of the coherence gate. By scanning the reference arm axially, and then by scanning the sample arm laterally, we can begin to scan our voxels in 2 dimensions or even 3 dimensions and start producing images. And once again, as with confocal microscopy, we detect singly backscattered photons from the optical section. Basal cell carcinomas appear as grayish, darkish nests, similar to what you see with reflectance confocal microscopy, albeit now they're at lower resolution and with somewhat reduced contrast. So OCT imaging begins to give us a good sense for the depth and the deep margins of basal cell carcinomas. And now the imaging helps us triage these type of skin cancers into those that may be superficial within about, let's say, 200, 300 microns. These tend to be non-aggressive. And so our physicians are choosing now not to treat these with surgery, but to treat these or remove these with non-surgical approaches such as laser ablation or topical therapy. Versus those cancers which are deeper than 300 microns, these tend to be typically the aggressive variety, the aggressive type, and these must be treated with surgical excision or with most surgery. Okay, so that's how the imaging is beginning to help us guide treatment. So we put together reflectance confocal microscopy and OCT into a single handheld device, and this is work being led by my colleagues in industry, Niku Sorefthimia and Bill Fox. And we've put together 3 of these handheld prototype devices, which are now being tested in the clinic by our most surgeon colleagues. And I'll show you an example set of images. This is of a superficial nodular type of basal cell carcinoma, showing you how with reflectance confocal microscopy, one can detect and diagnose the presence of these type of skin cancers. And with orthogonal and en-face OCT imaging, you can get a sense for the depth or the deep margins and the lateral spread or the lateral margins. So what's happening now is such combined RCM-OCT images are beginning to give our both surgeons 3-dimensional microscopic views of these kind of lesions that can help guide both diagnosis as well as treatment. And so my most surgeons have been busy looking at the role of RCM-OCT imaging to guide most surgery of complex skin lesions. For this type of basal cell carcinomas. In particular, one of our most surgeons, Dr. Chi-Shan Jason Chen, along with his group of fellows, have finished RCM-OCT imaging guided diagnosis, followed by imaging to look at the depth and the deep margins and triage for treatment, followed by the treatment itself, whether surgery or a non-surgical approach, followed by imaging to confirm clearance of these cancers on the patient, followed by imaging over 5 to 6 years to monitor the efficacy of treatment and to watch for any recurrences. And they have done this in 176 basal cell carcinomas from 114 patients. Very cool work, which is all unpublished. And in fact, they recently submitted this for review and for publication. Now, for the treatment of superficial types of these types of cancers, Dr. Chen's preferred approach for removing them is to do laser ablation with a pulsed erbium YAG laser. He asked me to share with you a couple of their key findings. One is that in nearly 40% of clinically diagnosed basal cell carcinomas, the imaging showed these lesions to be of the superficial kind within 300 microns, so he could treat them right away with his laser ablation approach. And so that's a good demonstration of a one-stop shop patient care paradigm. The patient comes in, you do diagnosis, you do treatment, and then you can send them home right away. The other result, which is actually pretty exciting and pretty interesting, is that in this particular cohort, in this particular study, the recurrence rate was only 1.7%. And this highlights the benefit of being able to use imaging to confirm for clearance, followed by imaging to monitor the efficacy of treatment over as many years as necessary. Typically, it's 5 years that they wish to monitor. Now, 1.7% is much less than the 10%, the 20%, the 30 or 40% that has been reported in earlier studies in which laser ablation had been performed, but there was no confirmation for clearance or no monitoring. So it's really exciting to think about the possibility that someday reflectance confocal microscopy combined with OCT imaging may help guide diagnosis, followed by looking at the depth and the deep margins and the type of these types of cancers, followed by treatment, whether it's more surgery or some other approach which may be non-surgical, followed by imaging to confirm clearance. And all of this will happen on the patient at the bedside in real time in a single patient visit. And then beyond that initial visit, imaging can be used for non-invasive monitoring to watch for the recurrence of any possible tumors and then treat them as soon as they happen. And such imaging approaches may serve as a very useful adjunct to pathology and in fact may even eliminate pathology in some settings. Very cool, what they've done over the last 5 to 6 years, my colleagues. Now, I said someday this may happen, but it's happening already in the hands of my most surgeon colleagues, as well as a few good colleagues in Europe, particularly in Italy and in Spain and in Germany. Okay, so this brings me to the final piece of my talk. I think all of you are still awake. I can see all of you blinking. But before I switch gears, I'll spend some time talking about new developments, new work that's in the pipeline, just to give you a sense for what's happening in the field, not just in my lab and not just at Memorial in New York City, but also with some of my other colleagues. But before I switch topics again, do we have any questions, any comments? And how much time do we have? I'll make it quick. This is quick anyway. All right, I'll keep going then. All right, just to give you a sense of what's happening in the field. So we're looking to combine wide field imaging with reflectance confocal microscopy all in a single device. And this is work being led by my colleague, David Dickensheets, and his group of graduate students, along with some of us at Memorial. And this is by introducing a tiny micro camera inside the objective lens of one of our confocal microscopes. We've shown feasibility with one of our benchtop setups. And what we're able to do now is produce these wide field images surrounding the exact location at which we could collect confocal images and image stacks. So now with this approach, our physicians will know exactly where they are within a large skin lesion when they do imaging on patients and they decide to collect images or image stacks or videos. So the wide field imaging approach can navigate for the positioning and the locating for RCM imaging. The next step is to advance this work from the bench into the clinic. And in progress is the building of a whole new lens with an embedded micro camera, as well as a whole new handheld confocal microscope. And this work is being led by Bill Fox at the company. And over the next few months, we expect to deliver this to our clinicians so they can begin testing on patients. In other news, we've been looking at machine learning-based algorithms, trying to develop these as well as to see if these can be useful for image analysis. And this is work being led by Kevanch Koze in my group, along with a group of machine learning faculty at Northeastern University and their graduate students, as well as Christy Fox from the company, and Melissa Gill is one of our dermatopathologist colleagues. One algorithm is for the detection of the dermal-epidermal junction in stacks of RCM images, with the idea and the hope that we may be able to standardize the process for image acquisition and automate that process. And the other is for the detection, segmentation, and classification of cellular texture patterns in RCM mosaics of melanocytic skin lesions. This is using a supervised approach called MedNet that was devised by Kevanch Koze in my group. And this is a multi-scale encoder-decoder network, which in a sense mimics how pathologists read pathology, which is on different scales with different resolutions. So the last couple of years, we put both of these to initial clinical testing. One was a recurrent neural network for detecting the dermal-epidermal junction. One was Kevanch's supervised approach for detecting 6 different types of cellular patterns. And one was another weakly supervised approach for detecting atypical patterns. And much of this work was done by graduate students at Northeastern University. So in one retrospective validation study, we found that we could detect the dermal epidermal junction with median accuracy of about 3 Z-steps. That's about 15 microns. And Kivanch's approach performed with moderately good accuracy for detecting benign patterns in benign skin conditions, but then the accuracy became much lower for detection of atypical patterns. And this makes good sense because benign patterns in benign skin conditions tend to be more uniform. They tend to be more homogeneous. They tend to be easy to detect and to classify. But the same cellular patterns that look so nice under normal conditions begin to look heterogeneous, they begin to look variable, they begin to look complex, and so they're not so easy to detect and to segment and to classify. And again, the weakly supervised approach performed with moderate accuracy for detection of atypical patterns. And then in a follow-up prospective clinical study, we found that we could detect the upper boundary of the dermal epidermal junction with higher median accuracy and higher repeatability than that for detecting the lower boundary. And this too makes good sense because as we image from the upper layers to the deeper layers, if you remember what I'd shown you about the axial response function, we start losing optical sectioning and we start losing resolution. And so it becomes more difficult to work with images that are deeper in the skin. As well as, once again, the weakly supervised approach performed with moderate accuracy for the detection of atypical patterns. So all in all, not such great results, because this highlights the challenges of trying to apply machine learning to these kind of images, which are really quite complex in their patterns and in their cellular and textural patterns. So there's a lot of more work to be done going forward. In other news, we're working on a high-speed video mosaicing approach. Again, work being led by Kevanch Kose with some of our colleagues at Northeastern University. And this is using optical flow, which is the flow of pixels between frames in a video, combined with a bit of deep learning. And what I heard from them recently is that they can process these now at 10 frames per second, which is the speed approaching real time, to begin testing on patients. The next step is to implement this on one of our handheld microscopes with the idea that we may increase the field of view, as well as, especially for mapping of margins, that can help guide our surgeons and can help guide surgery and treatment. As well as, we're trying to see whether we can apply a deep learning approach to detect the margins of lentigo-malignant melanomas in RCM video mosaics. And Annabelle Alfonso in my group had been looking at a number of different approaches, and what she's seeing is moderate accuracy for the detection and the classification of margins. So again, this is all early stage work, a lot more work ahead of us. So with that, I'll say my thanks to Memorial for providing me with the seed capital to establish my optical imaging lab and my program, as well as my thanks now to the NCI, the NIBIB, and the NIDCR for being tremendously supportive of all of my work. And since all of this work has now been fully funded by the NIH for many, 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 many years, I thank you all for your money. And thanks to my wonderful team of engineers who build devices, engineers who build algorithms, and our clinicians. They're just wonderful, our clinicians, our pathologists, and our biologists who do all of the clinical work. Thank you.
Well, we have time for questions. All out while we were taking intermittent breaks. I don't see any in the chat. Do you see anything?
There was a chat from earlier.
Yeah.
How does a confocal technique perform actually between lymphomas?
I think lymphomas are too deep for us to image, right, Jim?
Microsis fungoides.
Okay. So you can do this in mycosis fungoides. All right.
Yeah, excellent. So you showed that you have the supervised algorithm to reconstruct the figures. So these are like trained that you.
Yeah.
So how many training?
Yeah, it was.
How many initial data you need to see?
This was. So one challenge we have is that this is a new imaging technology, so we don't have large amounts of data for training. So this was a few hundred mosaics. And just to label those took us a good 3-4 years, just to figure out what to label. And especially these patterns are quite complex. But now that there are more and more groups doing imaging on a routine basis, my hope is that we can gather a lot of data And also the imaging is becoming a little bit more standardized. So I'm hoping that we'll be able to gather image stacks and mosaics from many different groups in Europe as well as in this country and in Australia, and we'll be able to build upon this work.
If you train with 1,000, then it's going to be getting more and more.
Yeah, for sure. Yeah. So one limitation. So the limitations are the same that, with any other approach, which is we have small amounts of data, not standardized, too many artifacts. So Kevansh Kode in my group has come up with an algorithm to detect noise artifacts and other artifacts, and to be able to at least figure out which are the mosaics that we should be using for training and which should be discarded. Yeah.
Question back? Yeah.
Yes.
So earlier on, you mentioned that Milind and can use as backscatter, like concentrate and allow us CD images, right? So I guess between different ethnic groups, do you see variance in the quality of images given different ratios of Milind, or
I'm sorry, I didn't quite catch the question.
The question is there different contrast with different ethnic groups, given that melanin is one of the sources of contrast?
It's, different levels of brightness and contrast, depending on the pigmentation level. So in general, if the skin is brown or black or has more melanin, or even basal cell carcinomas, which are not normally pigmented, but they're the class called pigmented basal cell carcinomas. So in all those situations, we do see variations. Yeah, for sure.
Are they significant between
They can be significant, yeah. Especially, in some cases, the imaging can be just saturated, and then it becomes difficult to image in skin that may have just a lot of pigmentation.
So one sidebar. So I was working on this at the same time Milind was working on this. I'm about as Caucasian as you get, and I couldn't see great images at all. Well, Milind actually put his arm in there, and he saw great images. So he was actually an incredibly good control for actually doing the development. So pigment helps.
Pigment helps, yeah.
Yes, go ahead.
Is there any benefit to imaging the same layer at different monochromatic wavelengths?
Yeah, that's a great question.
Repeat the question.
The question is there any benefit to imaging the same layer at different individual wavelengths? That's a great question. In fact, one thing that, if you think about me scattering and me backscattering, especially single backscattering, you actually can get modulation in your backscatter as a function of wavelength for a given scatterer. And so one possibility would be to use different wavelengths, different closely spaced wavelengths to look for a modulation, and you can then begin to get differential contrast for structures, something that we haven't done yet, but it's a possibility. You could use different wavelengths. In fact, Jim himself had put together a microscope with different discrete wavelengths, that can be used with the idea that you can look for differences in contrast. So possibly, yes. Yeah. The theory to back up that possible effort.
One more question.
Yeah, follow up about the training data. Are you able to work with doctors in the field getting the pictures and the results that they diagnose from those images, or is that some kind of patient privacy violation, or is there just not enough in the field? Yeah.
So generally when so the question was whether we can easily get images and data from our patients, or would there be any regulations that would prevent us from doing that? Typically privacy regulations. So the way we do our research studies is that we have to, in fact, consent our patients when we decide to collect, images for research purposes, as well as patients being routinely imaged will oftentimes agree for us to let them use their images. But we do have to get, the bottom line is that we do have to get a yes from the patient. And there are patients who might, who do say no on occasion. Yeah, that's always a big part of what we do. And the way what happens with clinical studies is that you design a clinical study and it goes through a process that's done by what's called as the IRB, which is called the Institutional Review Board, that reviews all clinical studies. They make sure that everything that you wish to do on a patient or with a patient is safe. And also you have to submit what's called as a consent form, which is reviewed. And once you get approval is when you can start doing the study, and you can start using these consent forms to talk to patients and get their consent, and they have to actually sign on these consent forms. It's a whole process that's in place.
Well, let's thank Dr. Rajadhyaksha one more time. Thank you.
Thank you very much.
And we have
Thank you very much. Have a good trip to India.
Oh, that was.
Thank you very much.
I think honestly, like.